Welcome back after the tea break, and still it's good morning. So I will continue what Umar has shown and what discussion that we had regarding part or panel discussion of Dr. Muhammad Ajmal, as he has clearly indicated you that we have to deal with biological data. So everything that we are dealing is actually biological in nature, be it a concept, be it mRNA or be it DNA. So we have to differentiate the type of markers that we are trying to catch for the precise diagnostics. And for acoustics, we have digital marker detection, and for MRI, we have imaging markers. And for biology or DNA, we are actually dealing with nucleotide. So for AI people who are not familiar with genome or genetics or biology, these are actually the features that we are dealing with. And in total, we have ACGT, four nucleotides that are making up codon. Now, what we are looking at is actually the codon and somehow decoding it using AI-assisted programming to catch up the markers, especially the biomarkers or biological markers. Coming to the test case of our early age disease, which is Alzheimer. As we all are aging, and we are also actually going to age with the passage of time, and whole world is actually aging, and so is our biological system. So if we look at the UN data that is the estimation of world aged population. If we look at it from the year 2015 to 2050, you can see that there is a paradigm shift in terms of long life. Why long life we are looking at? Because of the availability of resources, precise diagnostics, and also the treatment. The people are normally aging more, and especially the European countries that have adapted a healthy lifestyle, their death age has increased from 60 to 75 now. So, and it is actually not 75, it is more than 75. So it means that we are going to have a huge amount of people that can have age-associated phenotypes. And it is not only the phenotype of the brain, but different phenotypes that can affect different tissues of the body. And if we look at brain as an organ, it is also aging. Brain has to undergo developmental stage, and these developmental stage are affecting the well-being of a person more than unusually. So we have prenatal phase, we have teenage phase, and which is called as infancy phase. Then we have adulthood phase, which is from 20 to 40, and then old age, which starts normally from 60 and above. And in Pakistan, we have some kind of a psychological behavior that when we become 50, okay, we are aging. So you can think that as psychology is also having an effect on our brain function as well as development. So if we look at brain development, 20 to 40 are the maximum age where we function most of the time in our full capacity. If anything happens at that stage, or we change our environment, or make ourselves prone to different unfavorable conditions, we can develop number of brain-associated phenotypes, and the one that is a spectrum of phenotype is actually the dementia. Dementia is a mental incapability that actually progresses with age, and it affects daily. वो दो अभी हो गई कुछ और data. So dementia is the spectrum, a continuum, which starts with brain dysfunction, which can be mild, which is not affecting the normal body function, but to a severe, where number of disease become more prevalent, and that also includes Alzheimer. This dementia has been associated with number of condition or factors that can make it worsen with the passage of time. These include modifiable risk factors. And if we look at modifiable risk factors, it includes physical inactivity, obesity, unbalanced diet. You just name it, and every bad habit that we can adapt as an individual can affect our mental capability. Uh, and Alzheimer is no exception to it. Looking at the demographics or data of people living with dementia worldwide, and again the estimation from year 2015 to 2050, you can easily see that there's a shift from 46.8 million to 131.5 people living with dementia worldwide, which is a huge burden. And if you look at the region-wise distribution, Asia is among the one that have highest contribution in this percentage. And if we are talking about Asia, then of course we are part of Asia, the Pakistan, which is the underdeveloped country or low or categorized as low social middle income country. Uh, just looking at the death rates that have been calculated again by WHO, you can also see a gender predisposition. If there's a gender predisposition, it means that genetics is contributing to a gender predisposition as a vital component. Our genes or our DNA, being of a male or female, make us more prone to different phenotypes. And looking at this, in some part, you will see that female are dying more than male, and in some part, males are dying more than females. The common clinical features that dementia patient experience includes memory changes that disrupt their daily life. They also suffer from challenges in planning or solving daily life problems. They also have difficulty in completing daily life tasks. They have some psychological behavior as well, where they feel some kind of confusion and also trouble in understanding different things. And one thing or digital marker that Umar has covered in his talk is the words or speaking capability. It also declines with age. Uh, they also have some misplacing things behavior where they cannot decipher where to put different things. And it is again related to the memory loss. They cannot judge whether they should complete a task in which direction or in which dimension, or even selecting anything for their own self. It becomes challenging for them, and they also become because of these parameters, they become socially inactive, or they have withdrawal behavior, which is again a psychological and behavior, and it leads to mood changes in their behavior, which comes under psychiatric conditions of mood spectrum phenotype. So you can say that psychology, which is a brain phenotype, brain associated phenotype, as well as Alzheimer, they have an overlap. So it is good to have good understanding of selecting the markers for proper diagnostics, and we have tried to cover it in our study. What we did was taking the digital marker, we took their DNA and also the brain images. And working on this proposal, we try to classify the dementia patient as well as Alzheimer patient into different categories. Clinical diagnostics are required at this point, and we need to have a doctor, a trained doctor who can classify them into cognitively impaired person, mild cognitive impaired person, mild dementia, moderate dementia, and severe dementia. From mild to severe dementia is the one where most of the Alzheimer manifestation is taking place. The important thing in this subclinical classification is a class which is mild cognitive impairment, which is not affecting the daily life of a person. And this is the starting point of our research as well. One has already told you about different brain region and what type of cognitive functions they are performing. So looking at Alzheimer diagnostics, there are a number of diagnostics tests that are performed, which include computed tomography, magnetic resonance imaging, as well as positron emission tomography scans. The one that is readily available in all of the medical settings, or you can say a patient can have an MRI readily available instead of CT and PET. So we focused on this MRI imaging in our research. Then we are trying to couple it with the medical history, which is indicating involvement of genetic in the manifestation of the phenotype. And we are also taking digital marker, which are acoustic testing, that we are not developing a hardware, but also a software to detect it as a pre-symptomatic persons. Moving on with our research that we did on MRI, just look at this scan that we have obtained after machine learning. And the CNN model was the one which gave us more than 90% accuracy in differentiating the MRI images of non-demented person, which was of the control. The plaques are not visible over here. Now moving from very mild demented to moderate demented, you can see there is a shift in images or plaque deposition. This is somehow different than this one with clustering over here. And if you move on to the severe, they are clustered everywhere, or most of the part of the brain is affected. Why this research was important? By working on very mild demented person, which were not showing any effect of the phenotype, and which falls under mild cognitive impairment, we were able to catch the one that were not categorized before as mild cognitively impaired Alzheimer patient. We had only moderate to severe. But after collecting the data and working on MRI images, we were able to identify this mild cognitively impaired group. Interestingly, this group was also detected in control population. So some of the shifting from control population was also observed in our mild cognitively impaired person. So if we try to infer this finding, we were able to identify pre-symptomatic control population in our cohort of control as well as Alzheimer disease patient, which were classified only as moderate to severely demented Alzheimer. So it is good to go with the genomics. As you know that the protein that is making the cytoskeleton of the uh, of the cell, this protein actually, because of the degradation, is accumulating, as Omar has already shown you. So we can infer that there are some contributing factors that can be either genetic or environmental, and our genes that are playing multiple functions in the brain can contribute somehow to the disease manifestation in those patients that were mild, uh, mildly cognitively impaired persons. What we did was we again collected the data, genomic data or exome or whole genome data of those persons, and we try to do machine learning on this subclasses of Alzheimer patient. And the machine learning and the results that we obtained, Omar is going to take it further.
Assalamualaikum everyone. Thank you, ma'am. Moving forward towards the genetics methodology and how we trained our model on the whole genome sequencing data, we started by collecting our data from a reputed platform and the whole genome sequencing data. And we know that since genetic data is quite sensitive, we applied a series of genomic data preprocessing steps, which started by applying quality control first. This I'll go through all these one by one, and moving towards then the imputation, which is necessary as well, and then LD cloning, and then filtering our SNPs regarding the ones associated with the assignment. After all of this preprocessing was performed, we moved towards training our model further with the testing, and our final goal is towards deployment in our software tool, as Dr. Omer mentioned before. The data preprocessing. We sourced the whole genome sequencing data, and it comprised of all the chromosomes that are available in a human body, from ranging from 1 to 22, and they were provided in a genetic format, which is known as BCF format, stands for variant call format. We then applied all of these quality control, imputation, and LD cloning preprocessing steps, and finally filtering the features out which were associated strongly towards Alzheimer by sourcing the study. The final result we had were 32 features which were provided us in a blink format. This is also a genetic tool we use for data preprocessing. Starting with the preprocessing. First we applied quality control. Inside the quality control, our first step was filtering the data for the SNPs that were not available in more than 2% of the population. We used Splink for all of this, and removing them were necessary as they were not common. MAP, which stands for minor allele frequency. The parameter set here was 0.01, which indicated that we were removing our features that were not present in more than 1% of the population of our data, from our data. And then moving with the Hardy-Weinberg equilibrium. Now this is an equation, and more appropriately you can call it as a check. Our whole data was passed through this check, and it indicates if our data was genotyped correctly and the provided whole genome sequencing data is authentic or not. If our data passes the check, then the features and all of them are preserved, and if it fails, then we filter them out. Finally, we had to check for gender mismatch by comparing the reported gender from the platform we sourced our data, and the genetic data and the genetic gender which the data represented. Filtered one person out from this, and as their gender was mismatched. After performing all of these four of these quality control steps, our features reduced quite significantly. And then we move towards imputation. Now, imputation is quite necessary. Imputation refers to filling our genetic data, filling our missing genetic values. As we know that all of this whole genome sequencing and genetic sequencing is done using technological tools, and they're prone to making errors. Imputation generally refers to having a reference genome which is accepted worldwide, and it contains the data which is most commonly present in the population, and we compare that reference build with our data and fill the missing genotype values which is most commonly present in the population. We performed this on a web server which is known as Michigan Imputation Server. We used their compute services as they provided it on, and it was free as they provided it on their web page. And the reference panel we used was known as 1000 Genomes Phase 3. This is the latest phase of the reference panel available globally, and it keeps improving iteratively over the time. After imputation, we had a much larger data, and then we performed LD pruning. Now, LD pruning stands for linkage disequilibrium pruning, and it is very necessary as it helps us in reducing our features significantly by removing correlated and redundant features. Because if so that we don't have to deal with multi-collinearity later on in our model training process. Finally, after the whole genetic pre-processing and filtering our final features using by sourcing a study which was done before us, we selected a final number of 32 features which were related towards Alzheimer's, strongly correlated with Alzheimer's. The final data set comprised of 282 individuals, which were our data points, in which we had 155 cognitively normal patients, which were our control patients, and 127 patients which were diagnosed with Alzheimer's. The split is fairly good for our model, and we didn't have to deal with any sampling. We then split them into a ratio of 70% for training and 15% for validation and testing each. The models we trained were some of the state of the art, which include in the classification problem, which include logistic regression, support vector machine, random forest classifier, and a 1D CNN network because our data was tabular. The final data was in this form. We had a patient ID identifying each individual, 32 SNP features representing their genotype, which was encoded in the form of 0, 1, and 2. Now, this encoding is done from our genotype on basis that if the person's genotype was homozygous in the case of reference allele, we encoded it as 0, and if it was heterogeneous, we encoded it as 1, and if it was homozygous in the case of alternate allele, we encoded it as 2 respectively, which is the standard that everyone uses for genotype encoding. We had an age feature. As we know that age is a strongly linked factor with Alzheimer's, so we could not have discarded it. And a POI4 column which indicated the presence of E4 allele in our individual. A POI feature is genetic region inside our DNA, which is proven to be strongly, more significantly correlated with Alzheimer's. Changes in this region have been strongly linked towards Alzheimer's as compared to other genetic regions inside our DNA. And then a final label which indicates if the person was of control or Alzheimer's with 0 and 1 respectively. The initial results so far are not quite decent, but we're working on improving them. But even though from these results we can see the random forest outperformed the 4 models with an accuracy of 67%, followed by CNN with an accuracy of 63%, and then logistic regression and SVM respectively. The accuracy for CNN can be significantly improved if we can somehow increase the data set, but we're working on it. The confusion matrix. The confusion matrix was plotted by evaluating these trained models on our tested data, which was split up earlier. And as we can see from this confusion matrix, one thing was clear when we analyzed it, that most of our models were, although not quite efficient, but comparatively were more confident in predicting Alzheimer's control patients as compared to Alzheimer's patients. As we can see that most of the results are similar. 9 were misclassified as 9 Alzheimer's patients were misclassified as normal patients, logistic regression. Similarly, 8 in random forest, 7 in CNN, and 9 again in SVM. These were the results so far, and Dr. Humera will continue with our part. Thank you.
Thank you very much. Assalamu alaikum, everybody. Can I ask who among you is from AI? Thank God, only 2 people. Okay, I'm Dr. Humera Ayub. I'm working as Associate Professor at the Department of Biological and Health Sciences at Park Austria Hochschule. and I'm here since 2020. And before that, I was at COMSATS Islamabad. I did my PhD from COMSATS Islamabad, and then I served as assistant professor at COMSATS Hyderabad. Today, I'm very thankful to both Dr. Maliha Azam and Dr. Umar, who have invited me and given me a chance to come here and talk about the projects, my projects, which I've done in AI. One of them is on age-related macular degeneration, and the other is in glaucoma. So I do not understand AI a lot, but as far as I have understood it, it's a set of a combination of algorithms that help us to establish something, to make a tool, or to design a solution for our problems. And that algorithm or that set of algorithms should have two important things. Number one, it should think like us, think like humans. That is, it should understand our problem. And number two, that it should have a rational thinking. That is, it should be able to perform the solution for our problems. Now, coming to ophthalmology, as I'm working in the field of ophthalmology for the last 15 years, when we apply both of these things to ophthalmology, that is, it should act like us, it should think like us, humans, and it should find solutions for our problems. AI has really helped us a lot. Talking about age-related macular degeneration, which is a disease of old age in which a person is unable to see directly at somebody's face, or the central vision is lost. They cannot read, they cannot walk, their daily life is really hampered. So what we did in this project, which is still going on, what we wanted to do was to develop a tool, to develop an algorithm that can help the patients with AMD to find out their residual eyesight. बहुत सारे मैनेजमेंट वाले हैं, तुझे पता ही आईटी का बंदा ठीक कहते हैं बच्चे बदतमीज हैं बड़े। मैंने कहा नेक्स्ट सेमेस्टर में सीसीए का कोर्स है ना, ये थे मैन को सीधा कर तो पढ़ा लो। मैंने कहा मेरे पास दो वो वाले एक मिनट में पढ़ा लो। मैंने को कहा ऐसा क्यों नहीं तो पीएचडीआईटी वाले ऐसे दे सकते हैं। पीएचडीआईटी में से सारे आईटी के हमें पढ़ने चाहिए। ये वाला है ना तो आप लोग I guess. Yeah. I got background noise. Okay, so the objective of this project was to develop a trained model so that the patients with AMD can utilize their bachi-kuchi eyesight, the residual eyesight that they have. But fortunately or unfortunately, we did not have any kind of data present on the internet so that we could train our model on that. Therefore, first we had to develop our own model, and then we had to train it, and then we had to apply it on the patients. Currently, we are in the phase of developing that model, in which we will be using normal individuals producing an artificial scotoma so that it acts just like AMD in their visual field will be blocked by that artificial scotoma, and it will be able to, the tool will be able to identify where is the residual site, where is the patient, the person actually seeing. Maybe he has somewhere here, the cells are perfectly all right. And therefore, once we identify that and train our model, we will train the patients so that they learn how to use that residual region. For example, the person is not able to see here, the central vision, but his vision is lost somewhere there as well. But he has a little bit of region that has fine cells, and they can help him in visualizing. So our tool is going to help that patient first identify this region where he can see, and then train him to utilize this region to see further. So this was one of our projects in which we are utilizing AI. Our students, we have a specialized center there, Center of Excellence for Artificial Intelligence. So the students in AI are working with us in making these models, then training that model, and then we are going to apply it on the patients. So the second project in which we have worked on is about glaucoma. Glaucoma is a disease in which the peripheral vision is lost. In AMD, it is the central vision which is lost. And in glaucoma, it is the peripheral vision which is lost. It's a very complicated disease. Number one, the disease is known as thief of sight, which means a person doesn't get to know that his sight is being lost until a lot of damage has been done and blindness has set in. So what we really wanted to do with this project was to identify those individuals who are at risk of development of the disease. And we can identify this risk only when we have substantial data, and that is the genetic data. So here in this project, we were working on the genetic data. We collected a few samples and we did exome sequencing for them. And after the exome sequencing, we applied various different AI tools on them, which I'm not very familiar with. Maybe your questions, if you have any questions, Dr. Umar will answer them. We did data curation and cleaning. The raw genetic data and genomic data was analyzed. Data imputation was done. Then ultimately we found the clean and curated data. Moreover, feature engineering and selection was done. There were certain different features which were very important, such as Grantham score, phylopy, and other such predicting features, which could help us in identification whether the variant was pathogenic, increasing the risk of the disease or not. So these were different features that we selected. Then feature correlation matrix was done, biomarker filtering was done. Ultimately, data processing and machine learning models were applied to that data, in which the decision tree classifier, random forest classifier, SVM classifier, etc. were applied. This work has been published recently in 2025. If anyone of you wants to have a look at it, it is also present as an open source on the internet. So with this work, we were able to, although we did not have a very large or huge data to begin with, we had only 15 families to work with. So with this data, we are still able to identify the individuals who are at risk of the disease development based on their genetics. Well, how? Because all of this application has helped us in identifying those variants which are increasing the risk of disease. So now we are further working into this domain. Our AI students are further working into the glaucoma side, and they are trying to refine this work further so that we can actually identify the people who are at risk of the disease development. So currently these two projects are in AI. Otherwise, my whole work is on genetics. I'm an ophthalmogeneticist. So this is all about my work. Any questions if you have?
So extending the extending the application of AI in genome data analytics, you have already seen that whatever framework or whatever tool you use for machine learning, you actually need to identify not only the disease susceptible individual, but also you need to classify them as per their phenotypic manifestation. Can be at any type or at any subclinical class. Type means you have looked at ophthalmic data that is quite common. Why? Because it is causing irreversible blindness, not only globally, but also in Pakistan. And we are unable to pinpoint the individual that are suffering from irreversible blindness at early age of their life. So using AI, we can easily catch the ones that are prone to have the disease. And by doing so, what we are doing is we can provide them pre-manifestation, clinical manifestation diagnostic, and also we can prolong the manifestation of those individuals and somehow we will be contributing in healthcare. Dr. Ajmal will be actually concluding the talk after Umar will show us the hands-on related to this session.
Okay, everyone. Now that I explained all the theoretical part regarding the model training we've done until now. Okay. Seems clear enough. Okay. We created synthetic data first, which was of the type that we used for training our model and to gain our initial results. These were the dependencies and the libraries that we loaded, including pandas and numpy. Most of you were familiar with AI, will recognize these. sklearn and TensorFlow, all of these libraries were used for training our model and performing pre-processing and data analysis. Moving towards our data, we sourced this. The original the data that we got after performing all the genetic pre-processing was of this type. We had a patient ID identifier as I showed before from the presentation, and 32 SNP features. Now, the SNP features had each of the SNP features had a significant ID indicating them of the form of which was in string forms such as RS1, RS2, and then we had an age column representing the age of our individual, and in a POI column in which it indicated the presence of E4 allele in our individual, and our final label where one was for indicating if the patient was diagnosed with Alzheimer's, and 0 was for indicating control patient. Now, let me zoom this out so you can have a clear picture. Now, this graph shows our feature correlation with our target variable, which was diagnosing if the patient had Alzheimer's or not, which was our label column. And since this data is synthetic, so most of these correlation are not true regarding their relation with diagnosing Alzheimer's. But the original data if you use the original data, then it would show that a POI region will most probably have the strongest correlation with our final target variable, and helps most in identifying our Alzheimer patients, and followed by our SNP features later on. For the data preprocessing part, since we cannot use any imputer in genetics data for filling in our missing values, we had to check if we had first of all we had to check if we had any null values which were not which were 0 in this case. And even if there were, we would have needed to remove them, as we can't just impute them using any imputer of machine learning, because we know that genetic data is sensitive, and if we had used any imputer, then that would have not led to a good performing model. The pre-processing indicated separating our training features and our final target variable, followed by a train test split, which I told before was with a ratio of 70% training and 15% of validation testing each. This was our original class distribution. As we can see that both classes were almost equally distributed, so there wasn't any need for sampling to perform. Followed by scaling, we know that models tend to perform better on normalized data. So and we tested this out without using a scaler as well. And as it shows that the model which used normalized data performed better. This was, as I told before, we trained a number of models, including random forest, SPM, CNN, and logistic regression. on our test on our data, and suppose the whole training done. The results are following, which display the classification report for each model. Our logistic question currently is having an accuracy of 43% over here, because this data is synthetic and it is not the original data that we used for our initial results, which I displayed earlier. Random forest was having 43% as well. All of them were ranging in around 40 to 50%. And their confusion matrices were plotted like this, as I displayed earlier. This is for the synthetic data, so we can't really make any assumption because of this. As I just wanted to provide an overview of what our final data looked like when we trained our model and the initial results we had. Yes, this was all for the machine learning pipeline that we used until now, and we're focusing on improving the results. Thank you.
I think just to extend the discussion, Dr. Ashman will be having an overview of one of the sessions that we have, and we will be concluding our talk. If you want to add something at this point.
Let Dr. Ashman add and then we can.
Okay. Then it will be.
So, मेरा क्वेश्चन है, ये मल्टीपल प्रेजेंटेशन ये सारे मेडिकल वगैरह से रिलेटेड, तो एक चीज है कि एक्सप्लेनेबिलिटी फैक्टर को किस तरह हम देख सकते हैं। लाइक हमारे पास एक प्रेडिक्शन आ रही है, तो हम किस तरह पेशेंट को एक्सप्लेन कर सकते हैं कि ये एग्जैक्ट फैक्टर्स हैं जो आपके इस प्रेडिक्शन में आ रही हैं। लाइक अगर एक डिजीज प्रेडिक्ट हो रही है तो हम वो किस तरह एक्सप्लेन कर सकते हैं।
For explaining the outcomes, if you go through the literature, in some countries, when people are at risk, so most of them, they do not like to know we are at risk. So you can ask the patient, if in some places there is no rules and regulations regarding this, so we can just ask the patient if they want to know the risk, then we can tell them that these variations are related to the risk and you are at risk. But we should not, I think ethically it is not good to report exact variations. If you find exact variations and the individual is not patient and you are telling the patient that you will be definitely having the disease, then I think there are several psychological factors also, and individual might get the disease earlier. So it is better to report the risk variations, but not the exact disease causing mutations.
But I still, my question is that we have a prediction, like saying, so even if we don't want to explain it to the patient, we still, at least doctor want to know that these are the exact factors. Like we say that we have a multiple features. So which features actually are the main contributors for this prediction of the disease, so that we can actually at least treat those issues.
So if you reach at a stage where you know everything, that this is the disease causing variation and this is the exact treatment, then you can tell the doctor, and doctor can explain all the things in the medication, whole plan, how the disease will be treated. But if you are not still at that point, and you just know the risk variations, or there are some other variations, but patient is not currently having the disease, then I think only this variant should be reported. And exactly this
and just to extend the point of view of Dr. Ajmal, if you take the example of Alzheimer's or through this figure, what we did was we had control populations as well as disease population. For control population, we the MRI was not done as purpose for the detection of Alzheimer, but we were fortunate to get the Alzheimer of control population that were not actually diagnosed with Alzheimer. So what we did was we compared it with our clinically diagnosed patient. Now we came up with a group of at least, I think, 18 over individuals in the control population that made a separate cluster when we were doing the MRI based imaging and doing the machine learning on that data. They were showing plaque formation. So by using this data and showing it to the individual, that you have the chance of developing Alzheimer's later in age, but what you need to do is you need to actually back further back. No. You need to actually work on modifying these factors that are modifiable, that are in your control. You can improve your health by adopting healthy lifestyle activities that include like jogging, walking, and having a good diet. Good diet is must. Why? Because one of the contributing factor for Alzheimer and plaque formation is oxidative stress. So you can easily take antioxidant diet to improve it. And you will not be having Alzheimer as detected through our machine learning that you will be having Alzheimer, okay, at even 50 years of age or 50 years. And maybe you will prolong the onset till 70 or 80 by just modifying these factors. Right, sir? Yeah.
I have a question.
Gigi.
As maybe I have missed that thing, but सीखिए जो जितनी भी प्रोडिक्शन है वो सीएनएन मॉडल के थ्रू हो रही है जैसे कि हमें पता है कि जो सीएनएन मॉडल है हमारा दैट इज अ ब्लैक बॉक्स बेसिकली अगर इसमें आपने एक्सप्लेनेबिलिटी का फैक्टर ऐड किया हुआ है कि हाँ हमें कैसे पता चल सकता है कि जो चीज हमारा सीएनएन मॉडल प्रिडिक्ट कर रहा है दैट वाज एग्जैक्टली रिलायबल कि हम उसकी बेस पे
Because I think Umar can explain it better.
Yeah, okay, I would like to add to the question of Mr. Wes and you. So I think that's probably the same question you are having. So that is like how we can say that why the model is performing this way. So this is a why question. What is the mechanism that has been followed by a model to predict a certain case? So one thing that we have discussed in our earlier in the session, that was feature importance. So we are having n number of features. So there are some concluding features. So we are scoring those features that what is the most prominent feature which is comfortable in predicting the model as healthy control or as a demented patient. So one thing is like feature importance. Those features are responsible in predicting the case on the individual level. So first thing. And the second thing is like this thing of the explainable AI that comes in the post hoc analysis. So there is a post hoc analysis that refers you that which model is behaving and how it is behaving with respect to the features that you have selected. So that actually comes in the post hoc analysis, and we are trying to enhance our work towards that post hoc analysis in our upcoming work.
So any questions, any further questions? Yes.
is dementia reversible according to you?
Is dementia reversible? Yes, this is the question.
Dementia is reversible. Yes, you can make it reversible. As you age, you are progressing. But you can have a healthy old age instead of having a bad old age. So what you can do is you should work on modifiable factors to improve the onset and somehow restrict the onset of disease. Maybe you never know. You will prolong the onset and you will actually adding up to the longevity in your aging. So it is vice versa. You have to work on modifiable factors to improve the onset. If our tool has detected some features that are making you prone to Alzheimer, not only the features can be of DNA based, but also MRI based and also acoustics. And acoustics is very important if you try to couple it up. Why? Because if I'm talking, maybe I am somehow having Alzheimer feature because currently I am having some kind of dementia because I'm forgetting. So it is possible that I will be getting dementia, mild dementia, but I need to have a proper screening tool for myself to identify the susceptibility for myself for the phenotype that is expected to come in during later ages of my life. So yes, of course, you can deploy all these modalities, and by getting the prediction, you can improve your lifestyle, and by doing so, you can restrict the onset of the phenotype.
Ma'am, can you explain that basically जो हमारे आपके के सिटिंग सिम्टम्स हैं जो dementia या Alzheimer's के वो वेरी करते हैं पर्सन टू पर्सन.
बेटे वो करेंगे. Why? Because हमारा जो genetics है ना वो personalized है. Everything is now merging to personalized medicine. Why? Because my genetic is playing different role than yours. So what we need is we need to have a living markers, I will say, that can differentiate me from you or from any other over here.
I would
So
also like to add one thing here. It has a lot to do with your lifestyle. So things that you eat, the way you live, what is your circadian rhythm, how much do you sleep, all of these factors affect the way you will respond to the disease and the way that somebody else is going to respond to the disease, along with the genetics. Genetics is just going to increase the risk or give you, put you at risk of getting the disease, but all this, these rest of the factors are the ones that are going to put somebody in hard waters.
you can say that you are doing risk prediction and improvement, risk prediction and improvement. Reciprocal relationship between the
So
two.
just like diabetes, people, if they have a risk alleles and they are managing their diet and everything, then they can manage the even diabetes also. There are many people who have normal glucose levels by modifying their lifestyle.
Yes.
Yes. My question is more towards the AS side. So you said that you are taking like multiple features while making the prediction, like the noise and stutter and jitteriness. So how are you guys like paying the weightage? Like how are you deciding that which factor to look into more, or which factor has the highest importance while making a decision? Is it via like assigning weights to certain classes, or what is the criterion for deciding okay this factor will be taken into account while making a prediction and it has the highest weightage compared to other factors when making a certain prediction for a certain patient?
Yeah, so first thing is that we are not taking noise as a feature, so we are removing the noise.
So it's a loudness.
Loudness, yes. So first thing can be done to recognize the importance of the feature that we have seen in the hands-on, that how we can score the features in terms of its significance in contributing or in predicting some case of healthy control and Alzheimer. And the second thing is like there is a post-hoc analysis. We have just talked about this, that why the algorithm is performing in such a way that even we have selected a number of features to predict healthy control or Alzheimer, still different models are performing differently. Okay, so in that case, we can run a post-hoc analysis so that we can understand why a certain algorithm is picking some of the features, not the others, in predict healthy control or Alzheimer's. So that could be done for experimentation.
So like you said that it will be taking into account which feature is important. So will it be personalized for every input for every
Yes.
patient?
Yes, we have just discussed, yes. So basically there is a generic set of the features and then there is a personalized set of features. Okay, so that is dependent on person to person because genetically we are different. So we are having a different code, we are coming from a different environment, we are coming from different culture, across the culture. So that is having a differentiation already inside our DNA. So yes, that is going to be a personalized treatment plan for each individual.
And so the next question, slide you mentioned that you took that English data set and then you I guess passed the Urdu linguistics and made certain predictions. So my first one is how accurate were the results? And secondly, was the model fine-tuned on certain data sets or was it only trained on English and Urdu was passed on the different style?
Yeah, so we did not fine tune the model, so it was a local training. Okay, so one of the thing that is differentiating between the languages is like the acoustic features. So we are having a set of features that is same across the languages. So even in the English language or maybe Spanish or Urdu language, they remain the same. Like the pauses, like the intensity, frequency. So that is having certain things that remains the same across the languages. So that is the, you can say that overlapping between the data set that the model can be trained on those features only, not like the features that are differentiated. So that is like a unifying factor across the languages.
You said that you in the sixth semester, we were dealing with the filler words and stopping words in the previous time. So, what are the filler words exactly? Like certain people, uh, I know someone he talks and at the end of every sentence he says Hannah, Hannah, Hannah. Will that be considered as a filler word or stopping word, or like certain people when they talk they say or take longer pauses for this
Yes,
particular?
yes. So basically, the spontaneous speech that has been converted as a transcripts. Okay, so when we are talking about the linguistics, we are trying to catch the words in terms of the language. And the stop words like the commas, stop words, and then we are having the filler words like and you are taking more pauses. So that's actually referring to the filler words. So in linguistics, when we are talking about the transcripts, we are taking the linguistic features. That is the pauses, number of pauses, speech rate, and filler words. Let's say these are three features. And when we are talking about the spontaneous speech, we are taking the intensity, that is the loudness, the spectral features, and the frequency features. So there is a differentiation between the physiological features and the psychological features.
And adding on to the Umar's point of view, the personalized features are actually helping us in identifying the subclinical class of the phenotype. So somehow we cannot disregard those features.
Yes, please say.
So please correct me if I'm wrong, but from what I understand or from what I read is that when we talk about the filler words and the certain words, they generate certain waveforms or the frequencies or when we convert it into the waveform graph like we showed spectrogram. So if they have a certain signature that is same and it is said that it is same across different linguistics, different languages. So instead of going for the linguistics part of thing, what if we only taken into account the waveforms and then decide whether these are the filler words or stop words. In this way, maybe we can deal with the issues of low resource languages like Urdu or the regional languages like Saraiki or Punjabi. So can we like take into account only the waveform and spectrogram or MFCC values and don't
Yes.
take into account?
So this is what we have done. We have only considered the spontaneous speech. We are not considering the linguistic part right now. So multi-modality that we have discussed is the spontaneous speech. For the hands-on to introduce you like the linguistic features, that was the part. But our model is trained on the spontaneous speech.
Basically, you're only taking into account the
Yeah,
waveform
because
and inside.
Yes, the waveform is the only thing that we are trying to predict.
Few people have speaking issues, like naturally they are not so conversational fluent by speaking. Will this model actually work for them as well, or marking them as
Yeah, we were just talking about like the people who are disabled, who cannot speak or cannot express their word. So what we can do, we can try to develop a model that can detect the sign language. So there must be a cognitive decline in the sign language as well. So that can also work in that case.
And actually genetics can help in identifying because the genetic, there is susceptibility for stuttering regarding genetics. And using genetic as one of the screening marker, we can use the stuttering being genetically associated and then it will be completely gone from that point.
So that's actually the benefit of having a genetic model as well, that we are getting the inputs of genetics data as well, and what is happening on the genotype with respect to the stumbling and the.
Or maybe some comorbidities that we need to look
Yeah.
at. There are a number of comorbidities that Alzheimer patients or dementia patients are having because it is an old age phenotype. So you have to extract the already associated strongly genetic features with the respective phenotype and then train your model to the remaining one. This is what we did for the Alzheimer, where we took the strong contributors for Alzheimer and focused on that first.
Yes, why can't you do it in terms of writing? Because I think writing also does affect, like the emotional effect, right? Depends on the pressure, depends on how it letters.
So the motor functions can also be predicted.
Yeah.
That's what we would idea.
Yeah, that comes actually under dyslexia and learning disabilities. Learning disabilities mostly start at early age of life when you are going to school and you are starting your education. So this is the point where you feel that, okay, there are some individuals that have difficulty in having learning in writing, learning in listening, and also replying to the listening of the teachers. So they are called as dyslexic, and it is an early age phenotype. So considering this as a strong contribution of early age phenotype, this indicator, we can disregard this as a contributing feature for Alzheimer, because when cognitive decline is there, of course, they are going to have it, but not all of the age patient will experience, or dementia patient will experience this. Mostly for the ones that have an early age, autistic and dyslexic patients have that and genetic, there are genes that are associated with these clinical manifestations. So we can again disregard those genetic markers, and then we can move on to the other if we try to take it as one of the feature. It depends on the patient pool that we will be having and displaying the similar trait and characteristics regarding Alzheimer or dementia.
So what you're trying to say is none of that can happen unless the person had dyslexia when they were born.
Yes, maybe after treatment, it was somehow corrected for them, and later age they can develop. But in order to have a machine learning on that regarding the caustic, regarding the genetics, you need to have a patient pool displaying the similar clinical features for that, or you can say similar clinical symptoms. And then you can make a separate pool of patient and you do machine learning on that. That will be a complete separate study, but you need to have the complete information of their childhood behavior as well as the old age behavior.
So in terms of AI, it depends on the availability of the data. So if the data is available, then the prediction can
Yes,
be done.
then
So
you can-
the data is like you cannot say anything.
So any further question?
No more
For the ones that are present online, you just drop the question in the chat box, and we will try to reply to you through the email or any mode that you think is suitable. We will also share our emails, and you can contact us.
Or we can have like 5 minutes to see the question in the chats so that we can answer it.
Manan, are there any questions?
Chats. I think chat is disabled. आप भी कर सकते हैं chat.
They can access. They do have access.
Okay, so if the people who are online, if they are having any questions, they can put it on the chat.
Nothing else popping up? So let's conclude.
Okay.
Ajmal, to you.
Yeah.
So I hope everybody has listening these important and very talks about Alzheimer's disease and neurological disorders. So and I think we should join hands, especially the clinicians, the computer scientists, the geneticists, to solve the problems of patients who have developed these disorders or who are at risk of doing this. And along this, we also need to have some patience also, because patience is the key to success. So if you are doing something and you are in a hurry, then you might lose something that is very important. So as Allah has said in the Quran, in Allah Masabirin. So when we are patient enough and we are working in a very good manner, then I think we can help the patients and we can help the humanity also. I think with these words, we should conclude this session. And inshallah, in future, we also be conducting these sessions so that our students can get the knowledge also about this. And patients, those who are waiting, that there should be some remedy for their diseases. There are some patients who are directly contacting me when they have, one patient is having eye diseases. And he is in my contacts also, and he asked me every time, is there any gene therapy available for this disease? So we should work hard to help those patients who want to enjoy the life. Thank you very much.